Okay, hello, and welcome everybody to June's second WESTalks, the Water and Environment Student Talks. As always, WESTalks is co-hosted with IC Impacts and UBC Future Waters groups out of the University of British Columbia. The organizing committee is Abhishek, Lili, Fuhar, Jaskran, Kelsey, myself, and Faria helps with the back-end stuff like getting the videos onto the WESTalks videos recordings onto the website. So thank you to our team. Here's the global map. If you've been around for a while with WESTalks, you've probably seen this before, but this is where everybody in the world has checked in from, and I will post the link to that in the chat in a second. So if you haven't in the past, please do add yourself to the map. There's a little red plus sign in the top right corner usually. And if you are truly calling in from Antarctica, please add yourself and then message us, and we'll give you a big shout out in the next session. All right, here is the spring and I guess summer, now that we said we're in summer, the summer 2022 lineup, and we are right at the end of it. Oh, we're right at the bottom of the screen here. It didn't look like that when I was editing the PowerPoint. With Dr. David Weissbrodt from TU Delft, Delft. And so that's June 2nd today. And so I also wanted to put in a reminder for everybody, you've probably been getting my emails about this, but next week is the WEST conference. So if you've loved the WEST talks for the past 2 years, our seminar series, please join the WEST conference next Wednesday, Thursday, and Friday. It's all virtual, free to attend, everything about water. So from engineering to policy to forestry, earth and ocean sciences, everything about water is being discussed at the WEST conference. We have just about 200 people registered so far, again, from many universities and countries around the world. So I can also post a link to the west-conference.ubc site where you can register for free. All of our previous WEST talks and this one in the near future will be posted on our IC Impacts WESTalks YouTube page, so go check that out if you have missed any of them, or you want to share WESTalks, any of the previous ones, share with colleagues. So on to today's speaker. So Dr. David Weissbrodt is an assistant professor at TU Delft in the Netherlands. He benefits from a strong interdisciplinary curriculum in chemical engineering for life sciences, environmental science and engineering, and biotechnology and systems microbiology. And we're going to hear about a couple of those things today, I think. His academic track feeds on achievements in microbial systems, process engineering, water, energy, and health sciences. And he also has experience in the private sector. He's an active at international levels with a professional service to several societies. Along with Margit Oosterkamp, he chaired the MMWWRR 2017 Symposium on Microbiological Methods for Waste and Water Resource Recovery. As well, he chaired with Merle de Kroop for the IWA Biofilms Granular Sludge Conference in 2018. David received the inaugural IWA MEWE Early Career Researcher Award from the specialist group on microbial ecology and water engineering of the International Water Association in 2019. And he was awarded one of the Best Teacher Awards of the year in 2017-18, as well as 2018-19 of the Master Track in Environmental Engineering. David's work in science is intimately linked to teaching for the Life Sciences and Technology and Civil Engineering and Geosciences program at TU Delft. All right, lots of acronyms in there and in quite an established career. So with that, I will stop sharing and hand it over to Dr. Weissbrodt for today's presentation. As always, any questions you have throughout the presentation, please post them in the chat box at any time, and then I think Lili will get to those at the end of the talk. So over to you, David.
Thank you. I'm trying to share the screen. Can you see the slides?
Yep, looks good.
Okay, great. And the sound is fine as well. Okay. So thank you so much, Carl and Leili, for this kind invitation to participate to your WESTtalk seminars. I'm honored to close your academic year of seminars. As you mentioned, I'm an assistant professor at TU Delft. I come from Switzerland. I have a Swiss-German name with a French-speaking accent. And as we discussed before, I'm very well connected to colleagues in Canada, from Montreal to UBC, Vancouver, via also Central Canada. So it's a pleasure to be in contact back with the Canadian audience and other colleagues maybe across the world. So with this talk, I would like to guide you through how we can harness microbiomes or microbial communities to valorize extracellular polymeric substances from wastewater. Likely, you know very well that microorganisms can be used to treat wastewater, like carbon, nitrogen, and phosphorus nutrients, maybe other more complex contaminants as well. We can also use microorganisms to valorize biomaterial. Here, it's an example of a wastewater treatment plant in Europe, in the Netherlands, called Harnasch-Polder. It's one of the biggest wastewater treatment plants in the Netherlands, around 4 million person equivalent. Likely in Canada and other regions, you have even bigger wastewater treatment plants. These are the biggest plants. And if you look at this image, you see the connection between environmental ecology, urban ecology, and potentially industrial ecology at the waterway. And we will see how we can actually connect these different dimensions of ecology from the environment to urban cities and industries via microorganisms. The aim is to see and to understand via this lecture what is the role of water and waste resource recovery facilities in the circular economy and how we can use organisms in this context. So wastewater treatment plants are reconsidered as water resource recovery facilities or water resource factories. Some colleagues in North America at WEF are given this acronym on water resource recovery facilities, and colleagues in Montréal and Laval speak about STAR, Station de Récupération des Ressources de l'Eau, in French, and it's an acronym that is always more used in the North American environment, and that's coming also into Europe as well. When we look at this wastewater treatment plant here, it's an example of a small plant in Switzerland and a bigger plant in Austria that has the same capacity as the one in the Netherlands that I showed you before in The Hague. You see the massive amount of wastewaters that need to be treated per day, and it's even more probably in Vancouver. You see the massive amount of organic matter that is needed to be treated to 200 tons of organic matter per day in biological oxygen demand equivalent, 40 tons per nitrogen per day, 6 tons of phosphorus per day that should be treated in this plant in Austria. You can see the equivalent of chemical energy that is contained in these wastewaters and how much biomass is produced while treating the organic matter, nitrogen, and phosphorus. So we can think how we can not only treat the wastewater, that is mostly the most important part, so public health and environmental protection, but also how we can valorize these massive loads of organic, nitrogen, and phosphorus resources while safeguarding the water systems, recovering water as well. So using microbial communities or microbiomes, the assemblies of microorganisms, we can see how we can treat the nutrients, but how we can recover nutrients and produce potentially high-value compounds as exopolymers that I will speak about today. At the same time, wastewater treatment plants, if they want to add advanced treatment to remove micropollutants or microconstituents via ozonation of activated carbon, you need to integrate new processes. So you need place on the wastewater treatment plant. So you need to intensify the wastewater treatment plant processes. And there are some ways to intensify these processes using biofilms, and I will come to that afterward. So this bioprocess intensification is very important for process integration. And we use microorganisms to intensify bioprocess by producing biofilms or granular sludge. Then these microorganisms can be harnessed for their metabolism, for their functionalities, to not only remove nitrogen, phosphorus, and carbon, but also to try to recover this carbon, for instance, as polydioxyalkanoates that are used for biodegradable plastics or extracellular polymeric substances that can be used as coatings and other applications that I will also explain from what some colleagues are working on as well. There are a multitude of metabolisms possible in these mixed cultures, and our scientific way is to understand who is there, what they can do, who can we select for them, who can we select for their good activities that are beneficial for process design and operation. So if we overlay this microorganism with the wastewater treatment plant, here it's an example of a wastewater treatment plant in Switzerland, and in the background you have the Eiger Mountain, so the north side of the Eiger, probably you know that. So it's one of the best wastewater treatment plants to look at the Alps if you go once in Europe. You can also visit the ones in the Netherlands, very nice. So by harnessing microbiomes, we can valorize bioproducts, we can protect water and health, and for that we need to address microbiomes with systems biology approaches and mixed culture biotechnology. This is more engineering, so quantitative engineering, and more high-resolution analytics to understand these microorganisms and their metabolism. And the aim is to integrate this science into education, into technology and innovation, and also into policy development and incentives toward resource recovery. For instance, in Switzerland, where I come from, there are new incentives at the parliament to generate a new law for resource recovery from environmental systems. So taking again this figure from the wastewater treatment plant in The Hague, in the Netherlands, we want to make the link between what operators can control at wastewater treatment plant for nutrient removal and what we can understand in the lab via quantitative biotechnology and high resolution analysis at DNA level, but also at protein level, at metabolite level. And since operators are able to operate a wastewater treatment plant to control a plant for nutrient removal, we believe that in the future, we should be able to control wastewater treatment plants as water resource recovery facilities to see how we can control the processes to trigger a specific bioproduction, a specific production of the material of interest. If you want to use the microorganisms like in the lab here, we need to understand the diversity of these microorganisms. So this goes via microbial ecology principles. We need to understand their metabolism, as I mentioned before, so the ecophysiology, under which condition they activate their metabolism, under which condition they cannot produce, under which condition they can produce. They can valorize the nutrients as biopolymers. So one important task in my activity or interest, as you mentioned before, Carl, it's really this interdisciplinary that we can develop between science, engineering, and education. Between science and engineering with conferences, we have often a very good link between how we can translate or bench idea or bench findings into applications. Then it's also to think how we can also integrate this knowledge into our programs in life science and technology, in civil and environmental engineering, for instance. How we can bring back this very new knowledge back to education. Because in the end, with the young professionals, we should bring them from the classroom to the laboratory to think at bigger scale as well in a real setting that is really complex in environmental systems. So the aim is really to think how we can teach about recovered circular resources, and I believe that your seminar series is very well aligned in this domain. So the learning outcomes for today will be to identify what is the central role of microorganisms to extract resources from wastewater as biopolymers, extracellular polymer, to see how we can reuse principles of microbiome science and engineering in this context, which methods of systems microbiology and omics can we use to elucidate these microbiomes. When we use all these sequencing approaches, DNA sequencing, RNA sequencing, protein sequencing, metabolic mass spectrometry, and protein mass spectrometry and metabolite mass spectrometry, we generate such a big amount of data. The question is always how we can translate this big data in a good informational data for engineering designs. This is very important to always think about that. It's not just about data generation, it's using this data for a specific purpose in the end, notably in the context of open mixed culture biotechnologies to help the circular economy. In my research group, it's a group for environmental life science engineering. We are integrated in a section of environmental biotechnology here in Delft, Department of Biotechnology and Faculty of Applied Sciences. So with colleagues and with PhD candidates and postdocs, we work on nutrient removal, on exopolymer production, mixed culture fermentations, also capturing carbon, nitrogen, and phosphorus using phototrophic organisms like microalgae, also other types of phototrophs. We look at nitrogen cycles in bioreactors and try to understand the pathways. We also look at antibiotic resistances or xenogenetic elements like resistance, but also the fate of genetically modified materials in urban water systems. For the sake of this presentation, I will speak mainly at this interface between granular sludge here that is used to intensify nutrient removal and the production of exopolymers that we can recover from granules or biofilm systems. Let's speak first about how can microbiomes be elucidated and engineered, what kind of principles and methods we can use, and I will come then into the application for exopolymers. As I mentioned, we need to understand the microorganisms under metabolism. It's a recall of what I have already presented. Here you see this type of aggregates that are called granules. These are biofilms and particles that settle very fast, that are very dense, a lot of biomass, and it helps to intensify the processes. Instead of fluffy aggregates that settle slowly. So if you have fluffy aggregates, you need a bigger tank and bigger settling tanks to separate the biomass from the treated water. With granules, you can do the separation in the same reactor where you treated the nutrients because they settle very fast, and you can operate that in sequences, the sequencing batch reactor. What is important when you address microorganisms is always to think about what is the energy source, what is the source of electrons, and what is the source of carbon and nutrients in their metabolism. The microbial metabolisms are always driven by these three components: energy, electrons, and carbon. Either the energy is a chemical reaction or it's energy from light. Electrons, there should be always an electron donor that is needed to perform the metabolism, and there is always a need for carbon and nitrogen and phosphorus to synthesize the biomass and the polymeric substances of the biomass. Then, on top of that, we can use mixed culture biotechnology, so with bioreactors, well-controlled, and systems microbiology with all these sequencing approaches to understand how this microorganism exacerbate their metabolism under specific conditions in the bioreactor. We can use mathematical models then to predict what can happen in this system. And the aim is not just to generate the data, as I mentioned before. We should be able to bring all this data from reactors and from omics and bioinformatics back to the process. And this should go via discussion with environmental engineers and operators. We had once a review article with colleagues also from the wastewater treatment plant in Vancouver, Paul Cadota, who participated in a perspective article with us to see how we can reintegrate all this knowledge that we can develop from lab into wastewater treatment plants. In terms of principles, to design wastewater treatment plants, it's mainly about understanding what are the carbon, nitrogen, phosphorus sources, electron donor, electron acceptor, the energy source, is it a chemical reaction, is it light, and how biomass is produced. Based on that simple concept, then you can derive stoichiometry, you can derive thermodynamics and kinetics of microbial growth. And with stoichiometry and kinetics, you can design wastewater treatment plants. So kinetics would relate to the volume of your reactor, how fast the reaction go, will tell how big should be or how less big should be your tank. With stoichiometry, you know what are the ratio of materials that are needed to produce the biomass or to produce your polymer that you want to valorize. We don't need to have all these sequencing technologies to design processes. Process design goes via stoichiometry and kinetics, and it is assisted by mathematical modeling. Using sequencing technologies and mass spectrometry can help us to understand at much higher resolution what are the processes that happen inside the cell, while the engineer generally looks outside the cell. So it's a combination between what happens outside the cell and what happens inside the cell to understand the metabolism of this organism under specific reactor regime. So we can complexify these approaches in science to see how we can combine top-down design and bottom-up design. It means how we can start from a microbial community and to try to understand stepwise what is the activity of this organism, the red organism, the green organism, the yellow organism. In most cases, we cannot isolate the microorganism, so we have to think about approaches that can give us metabolic insights about this organism while still working with a mixed culture. And this can go via enrichment cultures of some organisms of interest, for instance, and to address their competition, their metabolism. If you have luck, maybe you can isolate the organism and try to understand their metabolism for each organism individually. How do they grow? How do they metabolize the substrate? How they can produce polymers potentially? And what happens if you have two organisms at the same time, or three organisms at the same time too? So from isolates, you can define co-cultures via synthetic ecology to address this relationship. So in the end, the science can go very deep by combining ecological engineering of microbial communities and synthetic ecology with starting from isolates and co-cultures. With all these technologies for sequencing, mass spectrometry, and all other microscopy methods and molecular biology methods, we can progressively shed light on microorganisms to understand which organisms are present, can we count them, can we get relative abundances in microscopy or flow cytometry. We are interested to get the informational molecules, so DNA or RNA, proteins, the metabolites, and we need a specific instrument to measure them through sequencers or with mass spectrometry. You see progressively the amount of data that you get from the system and what we can do from this data. Sometimes an experiment can last maybe 3 months. Then you get all this data. It takes you 1 year to process the data. Because you have to develop this bioinformatics skill set at certain time. So also in our education, we have to think how we can reintegrate this knowledge of biotechnology and also multiomics to better understand processes and what happens inside the cell. So from the genome to the transcriptome to the proteome. So all these metabolic regulation pathways are related to these molecules, these polymers like DNA, RNA, proteins, and the metabolites. If you only sequence the genome, you will lose all the information after the DNA. It gives you the potential. If you sequence the RNA, you only get information between genes and RNA, but you don't know what happens at protein level. You don't know if the protein was active. You don't know if you got an enzyme, therefore. You don't know the flux of metabolites. So ideally, in science, you want to bridge all this. You assume that if you want to do all this to address the process for exopolymer production, for instance, you get so much data, so much effort that you have to use a rational approach. But you can start with the genome, and maybe then you can sequence the proteome. It gives you more insight on the express functionalities of microorganisms. And from the reactor, you can get what happens at metabolite level in metabolic pathways. So in most of our labs, the discussion is how we can complement our strong knowledge on bioreactor and classical analysis with this high throughput metaomics. But this brings us to think about resources, so money, but personnel, but also instrument and square meters and skill set that we have to integrate into education. Definitely, the aim is not just to generate data as described here, is to move beyond the data to try to implement this data into a model that can be useful for design of a physical system in the end. But often between the science, in many studies, we stop at data generation level. In engineering, we are more at the physical system. We need to develop a language of love to get this model that is useful for both the scientists and the engineer. But these are the global principles that we apply in our studies on microbial ecology and systems microbiology in wastewater treatment and resource recovery. So let's try to see now how we can integrate that for the valorization of exopolymers from wastewater or sewage. So all the research that I will present now is linked to multiple interactions that we had in Delft with different colleagues, and also PhD, master and PhD candidates that joined our section, notably Nina, Lorena, Sergio, and Simon. And we have a strong interaction with Jumelin and Martin Lostrecht and other colleagues like Jure, Mario, and Steven Picken, who are colleagues in Delft, but also in Aalborg University in Denmark, like Rafael Nielsen, Max Albertsen, and Morten Duehold. And previously, I worked on granules and EPS as well at EPFL in Switzerland, in Christoph Rediger, some interaction with Thomas Noy in Germany, and the Remote Center for Environmental Research. And Lorena came from Brazil, working with Regina Costa in Florianopolis. So we had strong interactions across regions as well. And with other colleagues in Switzerland, like Eberhard Morgenroth, Nicolas Derland, and Ilona Schivak, in the characterization of these polymers. I thank them for all the interactions that we have. It's really rich across my curriculum to have worked with them and interaction with them. This is continuing. So let's start from granular sludge. I presented to you before these particles that settle very fast to intensify bioreactors. So this is a reactor that I had operated in the lab in my PhD. And you see these particles that are small stones of 1 to 2 millimeters that settle very fast in the reactor. There is no carrying material. So these are biofilms that are auto-aggregated. And let's dig into these biofilms. So if you do confocal laser scanning microscopy, you can see the cross-section of these granules at high resolution. And you see that it's a heterogeneous architecture with some compact clusters, likely microorganisms that assemble in microcolonies that are very compact, and maybe other materials or other microorganisms that are around. There are some channels as well. There will be some diffusion limitation for substrate, but also channeling of substrate across this architecture. So often in models, we consider them, we conceptualize them as onions with different layers that are aerobic, anaerobic, or anoxic. But if you look at high resolution, it's more conglomerates with different type of architecture. So this is already fascinating to see this assembly of microorganisms that are auto-aggregated. There is no carrying material. Here on this C panel, you have a comparison between the granule and the floc, and you see the difference of metrics between an activated sludge floc in wastewater treatment plant that settle very slowly and the granule. And there are different architectures of granules. So depending on the growth rate of microorganisms form different types of biofilms. So fast-growing organisms, they tend to form more cells that are suspended in the biofilm, let's say. So very homogeneous biofilm, as we see on this cross-section here. Slow-growing organisms will tend to form much more compact microcolonies, like this organism that remove phosphate from wastewater, called Acinetobacter. So it forms these typical conglomerates. You see a 3D architecture here of these granules. So granules are made of microorganisms, and there are also some organisms that we don't want. They are filamentous organisms, and they will lead these granules of flux to settle very slowly. You see these filaments here. And this is problematic for wastewater treatment plants. You don't want them because of the settling afterward that is impaired. So by granulating the biomass, you can increase the settling rate of the biomass, and you can also intensify your process. You can have much more biomass per volume of reactor. Therefore, the volumetric rates of conversion are higher. And you can have all the settling happening in the reactor if you stop to aerate the reactor, for instance, or to agitate. So we see here these microbial assemblies. On top of microorganisms, there are extracellular polymeric substances, or so-called EPS. Often you see that in literature. And this EPS, they breach the microorganism, they form the glue between the microorganism and these biofilms. And there are different types of constituents in this EPS. There are polysaccharides, lipopolysaccharides, proteins, enzymes, there is DNA or nucleic acid, other types of proteins with, are related to sugars, glycoproteins, and many other types of components like amyloids. So it's a zoo of microorganisms, and at the same time, it's a zoo of constituents. Microbial communities are very complex. There are functional exopolymers or functional extracellular polymeric substances are also very complex. And therefore, it's very important to interact between scientists and also with engineers to address this complexity. Only one skill set is not enough to address these complex microbial communities and complex assemblies of polymers. You need microbiology, you need chemistry, material sciences, engineering and molecular biology, many different disciplines, mathematical modeling as well. So here, for the sake of this presentation, I will mainly focus on the extracellular polysaccharides. Initially, it was postulated that these EPS are form polymers that are like alginates. It's not exactly alginates in terms of chemical compositions, but they behave like alginates because they can form hydrogels, like alginates that are used to encapsulate flavors in chemical engineering, for instance. So these extracellular polymeric substances, and specifically the glycoconjugates, the polysaccharides, are also very diverse. So with lectins, you can stain glycoconjugates in biofilms or granules. So these are cross sections of granules that I made that we analyze with fluorescent lectin binding analysis. With different types of lectins that are recovered from plant materials that will bind selectively to glycoconjugates. And you see these amino sugars here, N-acetylglucosamine, for instance, or sialic acids, alpha glucose, alpha mannose here, N-acetylgalactosamine here. So these are not just alginates with M blocks and G blocks. They are amino sugars, they are sugars, and other types of glycoconjugates. And you can map the microorganisms with stainings, for instance, and you can stain as well the glycoconjugates with these lectins, here displayed in green, and gives you these beautiful images of this assembly of organisms that are assembled using this glue of glycoconjugates. They are not only glycoconjugates, as I said before, there are also proteins and other nucleic acids, for instance. Using confocal laser scanning microscopy, you can co-localize the organisms, here displayed in red, like Aconibacter, this organism that removes phosphate, that was here hybridized with fluorescence in situ hybridization, and the lectin staining in green to display the glycoconjugates. And you see the organism in this matrix of polymers, or surrounded by this matrix of glycoconjugates here. Then the idea is to think, okay, we see this organism in this matrix. Is there a relationship between the organism that is here or here and the matrix that is around? Is it the microorganism here that is producing this matrix, or it's another organism that we have not identified that is producing it? So the question scientifically is to understand how can we capture the metabolism of Aconibacter, not only for phosphorus removal, but also for exopolymer production. So we need to make enrichment cultures of this organism to try to form biofilms like granules in the bioreactor, as we did here with Nina and Lorena, and you see the shape of the granules again here. And then the question is how we can extract these polymers that behave like alginates. If we contact this extract of polymer with calcium or sodium, they will form either films or micelles, let's say, or hydrogels. And you see progressively the material property of these exopolymers. So now our interest is to understand this organism more in detail, because it's important for phosphorus removal from wastewater. It can also remove nitrogen under certain conditions. It removes the carbon as well. And then the question is, on top of the removal of carbon, nitrogen, and phosphorus with this organism, can we also produce valuable substances like exopolymers? If you want to know more about extracting EPS, one of the former PhD candidates of Humelin and Mark van Loosdrecht in Delft, Simon Feltz, did a Jove article in the Journal of Visualized Experiment, and you can have a video online that you can download about his method that he developed to extract these exopolymers. He also tested different methods, and this is one method here where he used sodium bicarbonate, heat, and mixing to get the highest yield of EPS extraction out of the biomass here in milligram of volatile solids of EPS per gram of volatile solids of biomass. He compared this method with different other methods, and he really got the highest yield. And this type of method is even used now at pilot and on full scale in the Netherlands with the first set of plants that are needed, that are built to extract polymers from biomass recovered from wastewater treatment plants. Simon also developed a method using chromatography of monosaccharides to somehow sequence the glycoconjugates that are present in EPS, to sequence the EPS glycoconjugates. So it's a chromatography, and you can get peak of different substances. And if you have standards for specific sugars or amino sugars, you can quantify them and get a profile, concentrations, composition profile of this EPS. It's a bit like sequencing the DNA. Now you can also sequence the EPS. And the idea now is, based on sequencing DNA, we can get information of microorganisms. We can sequence their proteins to know as well about their metabolic pathways that have been expressed potentially. And with the sequencing of the EPS, with this chromatographic method, we can get information on the composition of this EPS. Could they vary over time? Are there always stable composition or not? So this is an illustration now on how, by understanding the microorganisms, by granulating and extracting the polymers, we can produce biomaterials for the circularity of the water chain, the water resource recovery facilities. Here, it's one of the first wastewater treatment plants in the Netherlands that implemented this extraction of exopolymers to produce these valuable biomaterials. And there is a second wastewater treatment plant in Utrecht, also in the Netherlands, that will open the second process at full scale worldwide next week for the production of this polymer that is called commercially now Caomera, via a consortium organized by Weil Haskoning DHV, a company, an engineering company in the Netherlands, the TU Delft, Chaincraft, and other stakeholders. If you think about the world market of alginate, I said that these polymers can behave like alginate. It's not alginate, but it behaves in terms of rheology and hydrogel production. For alginate, the world demand of alginate is around 40 kilotons per year. How much exopolymers we can recover from wastewater treatment plants? So this plant in Utrecht that will open for exopolymer production next week, it's around 500,000 person equivalent. This plant, we can produce up to 2 kilotons per year for this wastewater treatment plant. Then in this wastewater treatment plant in the Netherlands, there are 200 or 300 wastewater treatment plants. So you can rapidly make the calculation. 2 times 300, it's 600 kilotons per year. So you can meet definitely the world demand of alginate. It's clear it's not the same market, but if you think worldwide, the amount of exopolymers that can be produced, if you find a specific market niche for the product that will be valorized, it's massive, the impact that we can have with wastewater treatment plant for the circular economy. So you see here the product value of exopolymers versus PHAs, this biodegradable polymers and other fuels or chemicals or biogas that can recover in wastewater treatment plant and extract processes. So this shows the really the potential, and this is into application now in the Netherlands actively. What are the resources that you can valorize and how you can valorize the product? You can produce hydrophobic coatings of paper, was shown in water research or sustainable material technology paper by UBC and other colleagues from Delft and other partners. You can make flame retardants as well. You may also made some nice jewels based on cellulose recovered from toilet paper and an exopolymer that was recovered from sludge that treats nitrogen, so anammox sludge. And this exopolymer can be used as a binder to bind cellulose nanocrystals. And this is a very nice highlight to show what you can do with these polymers recovered from wastewater. And students from the TU Delft, from our program in life science and technology, actually won a European competition to develop water skin based on these exopolymers. Here you could see potentially an artificial leather that can be used to make shoes, for instance, or other types of materials. When I think about water skins, it always remembers me when I was a child of this type of system to drink water when we were with our school trip, for instance. And I could think, okay, maybe in the future we will see that these water skins could be produced based on these exopolymers that are produced from the wastewater treatment plant to contain drinking water. This is quite interesting to think about. So this is about the exopolymers, the biofilm, the granules, the exopolymers, and their valorization. If you want to understand more the microorganisms, so let's dig into the next 10 minutes into the metabolism of this organism. So the aim is really to understand this microbial community and specifically some organisms, like the one that remove phosphate, to see what is the metabolism to produce EPS out of the substrates that are present in the wastewater, like the organic matter and the nitrogen water. So we can clean the water, so we can recover a chemical resource at the same time. That is valuable for many applications, as mentioned before. To understand this ecosystem, if you do sequencing, you get a lot of data. You get all names of possible microorganisms. So at a certain time, I was thinking, oh, can we relate this microorganism to functionalities? And if you want to rationalize this information, you can try to build conceptual ecosystem models. There are some colleagues in Aalborg, for instance, that I mentioned, who worked on functionalities of microorganisms. They do ecophysiology analysis. And based on reading literature about all these organisms, you can try to think, okay, across all the pathway of carbon, phosphorus, and nitrogen removal, which organisms are involved in these pathways. So these are the pathways for carbon storage and removal, for phosphate removal, and nitrogen removal under anaerobic and aerobic conditions. Then we can also think how these EPS are produced. And actually, within all these sets of microorganisms that are detected with sequencing, which one are producing this EPS? Which one are the good organisms to select to produce this EPS? And for instance, this organism that removes phosphate, Acinetobacter, that I already mentioned, we are interested in digging into the metabolism of this organism to see, can this organism produce EPS while removing carbon, phosphorus, and potentially nitrogen? These conceptual ecosystem models are very useful for functional analysis afterward. So these PAOs, like Acinetobacter, they are in database. So in Aalborg, they have a database called MIDAS, the MIDAS field guide here, that give an information on all microorganisms from wastewater treatment plant and the potential functionalities. They are typically selected in sequencing batch reactor, where we alternate anaerobic and aerobic condition. In the lab, we can do in reactors of 1:1 height to diameter ratio, or in bubble column design as well. And they have the specific metabolism under anaerobic conditions. They store the carbon into polydioxyl canoates. Under aerobic conditions, they grow on this polydioxyl canoate. Then the question is how they will produce around the cell this EPS. So we did some experiment in this reactor to see when we were selecting for Acinetobacter, are they producing EPS? So we saw a selection of Acinetobacter, the PAO in blue here, and at the same time, an increase of the mass fraction of the EPS in the biomass. It does not say that this organism is producing it. It says that while it grows, there is production of EPS under the condition of the reactor. The question now is, are they involved in this EPS production? We can follow with FTIR spectroscopy the composition of the polymers at different time points. We can measure proteins, we can measure polysaccharides, calculate their ratio. And with the chromatographic method developed by Simon, we can analyze these neutral sugars and acidic sugars to sequence the EPS. So with all these methods, we can track the microorganisms, what happens in the reactor, we can look at what happens in the EPS. Then with genomics, we can try to isolate the genome from the organism of interest, as I did here, and after quality control and completeness control to look at the functional categories. And if you dig into the EPS, you will see that there are so many different types of functional categories and enzymes that can be involved in this EPS formation. It's not like just nitrogen removal or phosphorus removal, or where there are some known enzymes that catabolize the substrate. Here there is a forest of enzymes as well involved. So then the question is to understand the regulation of this pathway, to understand how this enzyme works, etc. We need to go via proteomics and mass spectrometry further. So here it's an illustration again of this complexity of the polymers between glycoconjugates, but don't forget about the protein. They form a kind of foam around the microorganisms that are surrounded by glycoconjugates. This was published by Sergio that I mentioned before, and this picture was taken by Thomas Noy in the group with fluorescence spectrum analysis and critical laser scattering microscope. Then Sergio worked further to understand some production of some substances in this EPS. There is a forest of substances, there is a forest of glycoconjugates. Either you can look at the whole, but it will end up in so long research to understand what is happening, or you can have a more targeted approach, for instance, on sialic acids that can be interesting to as they are used as pharmaceuticals. You can think, can we produce pharmaceuticals from wastewater in the end? And they're also involved in the regulatory pathways in pathogenicity between microorganisms. So Sergio tried to understand what are the metabolic pathways of these non-ulosonic acids like sialic acids, how they are formed, and what is the diversity. He looks at the metaproteome to understand, okay, what are the proteins, the key enzymes that are involved in these pathways. To look at the diversities of the synthesis of these sialic acids. So this pathway relatively complex, and in this paper that we published, you can have much more information about the pathway, the metabolic pathways from inside the cell to outside the cell, and how they release this component, how they degrade this component. So there are some transport production inside the cell, transport outside the cell, and consumption by other organisms or interaction with other organisms. So it's quite complex as well. So we can do some pathway level analysis. Maybe if you discuss with your colleague Stephen Hallam at UBC, then we get more insights on that. So Sergio developed a conceptual model based on his mass spectrometry analysis and protein analysis about the activity of different organisms like Acinetobacter, but also other flanking population like this Chryseobacterium and Clostridium that are present in the biomass. So Acinetobacter, based on the genome information and protein information, can potentially produce this sialic acid displayed in purple here. And other organisms can have enzymes like sialidase that can cut this sialic acid at the surface of the cells of Acinetobacter and potentially consume the sialic acids that are sources of carbon for their metabolism. Acinetobacter can potentially reintegrate it as well for growth. So in this ecosystem, it's a kind of ecosystem of EPS producers, EPS consumers, but the question is, are there specific consumers or not, or is it general decay? So Sergio is also looking at this. So in the perspective on what Sergio is currently doing now, some experiment with substrate labeling with C13 label of acetate that we feed in the bioreactor to understand the kinetics and the metabolic trends of EPS production in terms of incorporation of the C13 in the EPS. And if you stop the C13 supply, you go back to C12 supply to see how these EPS are turned over in this community. Is it a fast process? Is it a very long process? So we will know that in the next article. And so the interest is to see are the C13 products that are produced by Acinetobacter, for instance, are they reconsumed by these other organisms? Is it a specific consumption, or is it more general decay of the biomass, and any substances that has decayed from the biomass is consumed? So to close this lecture for this last 2 minutes, hope that I could give you some insights on how we can elucidate and potentially engineer microbiomes for resource recovery and circularity. The question is how we can use this data. So it's always about that when we generate data. So what can we do about this data? The main take home for responsible biorefineries at wastewater treatment level, we have to understand that microorganisms and their metabolisms are very important to understand for process design. It's not needed to have sequencing to design a process, but it generates all the scientific knowledge that will help better understand how we should operate these systems in the future, how we can trigger maybe the production of one product, like say sialic acid, or another product that can be very interesting. This data need to be translated into engineering concept. We need to develop a language of love between systems microbiologists and engineers. We need to teach that into our programs as well. We need to interact between specialists that are experts in each discipline as well. This multiomics science helps to crack the complex biosynthetic pathways of this EPS. And definitely, the aim is to develop a responsible science, engineering, but also education to foster a focused bio-based solution for the circular economy, protection of the environment, and the public health as well. With this, I would like to close this lecture. I hope that I was able to show you the link between microbiome science engineering and water resource factories, the link between the lab and the full scale. And definitely, if you elucidate microorganisms and their metabolisms, it will provide you the scientific insights to potentially valorize these polymers from wastewater. Thank you very much for this invitation, and I look forward for interacting with you further.